initiatives. I am Herbert Haubold. I'm the ILTER secretary, and my responsibility here is to administer the research initiatives. Our research initiatives program is a funding program for small research grants. So we fund rather compact undertakings in themselves, or for example, seed grants, which may lead to larger undertakings or connect other existing projects. It could also be thesis support and so on. So many different options. And with this, ILTER also supports some active research beyond the coordinating committee, but the coordinating committee is of course very interested in this. That's why we have linked this webinar with the coordinating committee today. And I'm really excited. If I look here on the participants list, this has really aroused quite some interest with participants from around the globe. And at this point, of course, I also need to thank our new IMO officer. IMO stands for International Management Office, Dishane, for organizing the whole matter. Now, this is the web address where you can learn about our research initiatives, about the program, its background. You can also learn a lot about ILTER. So I really invite you to visit the ILTER website and explore it a bit, and then also learn about the research initiatives. For now, there's only a very short section on it per project, but there will be more to come. Now, why are we doing this? ILTER wants to create some examples for use cases. So the prerequisite for each of our initiatives to be funded is that it demonstrates clearly how and why it exploits the ILTER infrastructure and ILTER data. So this is really essential. So any kind of research that is nice but could be done without ILTER just as well is not so much in our focus, but this should really be tangible use cases. And we are, since last year, applying a new and targeted approach where we have competitive calls for proposals via the website. So all of those who report today about their work have been evaluated, and it was a tough evaluation because we were overwhelmed by the huge amount of calls we got, responses we got, and we had to select a small number of proposals which we could fund, these 5. So all of these are really very good. They are evaluated towards the ILTER strategic goals, among which is of course the scientific excellence, which is very important. And speaking of scientific excellence, I will now hand over to Ellie Groner, who is the chair of the ILTER Science Committee.
Thank you. So we had quite a lot of nominees and quite a big challenge. And thank you very much for all the EC and Science Committee members who helped value the calls, and it was quantified and hopefully objectively assessed. This is not a regular assessment. We have to consider not just the science merit, but also the contribution to the ILTR community. And by that, eventually we found the project that we think are suitable, and hopefully we will have some very interesting project that will help establish the importance of LTR and how LTR is different to other research. And I hope that we will be able to learn from those projects, and the next time, and hopefully we will have those calls, more of them in the future, and we will be able to learn from those projects what works and what works less, and what contributes to our network more than the others. And accordingly, we might adjust the next call to try and address special areas and special directions and angles that we want to address. I think that's enough for talking around, and now we have to get to the science. Herbert.
Okay, thank you very much, Ellie. So then we will already come to our first one, and I welcome Simon Robson from the University of Queensland, who is going to present to us Microbat Monitoring for the ILTER-EAP region, where EAP is East Asia Pacific.
Thank you very much.
The floor is yours.
Thank you very much. Can you see that slide okay and hear me okay?
Yes, we can see the slide.
Fantastic. In that case, I will go to the start of the slides and we will be ready to go there. Okay, well, thank you very much for the opportunity to talk to you about this project. It's particularly nice to be able to put some faces to names. Okay.
Sorry.
Simon, we're seeing your presenter view.
How do I stop that? My apologies. Has that fixed it?
Yeah.
Great. All right. Again, thank you very much for the opportunity to talk about this project, which I'm carrying out with Will Edwards. So our goal is a fairly simple one. We'd like to provide real-time information for scientists and environmental managers in Southeast Asia about a group of mammals that are very important, but are relatively poorly studied. And these are the Microcoroptera, comprising approximately 20% of the world's mammals, and with one of the global hotspots in Southeast Asia. And there's some rather nasty predictions there. Bats are not alone, I guess, in how many species might be going. See, I'm not sure why that's sort of progressing. So the project, the goal is to provide real-time information. The project is supported by the lead ILTR leaders of Thailand, the Philippines, and Vietnam, and by Titley Electronics, a company in Australia that produces software and machinery for recording bat ultrasounds. And we're going to achieve these goals by using these newly developed acoustic techniques to allow continuously monitoring of microbat populations. I do apologize for that. And our plan is to establish monitoring networks in Thailand, the Philippines, and Vietnam as a proof of concept, work through the system there. And it builds on a recently established project running in TERN. So we're taking some of the lessons that we've learned from TERN and applying that to Southeast Asia. So where we are at the moment is the 3 ILTR leaders have selected at least 2 participants from each country, and they range in various skill levels and expertise with bats. We're having a workshop in January next year, hosted at a university and a field station at Vietnam. And at that workshop, I and representatives from Titley will be training those individuals in the techniques of bat call detection, analysis, and data management, which is clearly becoming a rather huge part. And then the hope or the plan is that individuals after this workshop will become proficient at detecting and identifying calls, the techniques that we use for analyzing data, making data available to downstream users, and predicting actual sites. And then those individuals will go back and deploy those systems in their respective 3 countries. And we'll see how it goes. So the newly developed techniques we're going to use is, there's been some great advances recently in techniques for detecting ultrasound. Titley are producing a new system called the Full Spectrum, the Ranger. It doesn't come with this beautiful bird, unfortunately, but you only get the recorder. You can now save up to 2 terabytes of memory, of rechargeable batteries. You can put them on solar panels. It allows you to record continuously nightly monitoring. And based on Australia, 2 terabytes of memory in an active area would allow you to record for at least 2 months continuously without having to go back to the detector. So that's very useful. We're also working on installation protocols. And there was an earlier mention of the TERN's biodiversity, the TERN's Threatened Species Index. We've been contributing to that as well. And we're great believers in the need for open access analytical tools. So the data will be open access to anyone, and we'll be providing additional software tools that people can use to analyze that, and hopefully possibly some computer front ends as well. We'll also be making use of existing resources, and there's some great ones out there. This is an image from CairoVox, which is a Southeast Asian library that has been set up. So this picture here shows you an ultrasound. So the y-axis is the frequency in kilohertz, and the x-axis is time. And these are 5 species of rhinolophids. I've picked rhinolophids because they're the really easy ones to distinguish. They're very, very different. We wish the whole world was full of rhinolophid bats. Unfortunately, it isn't, but that's okay. So we'll be making use of it. We've chatted with CairoVox, and they've very generously made those calls available to us. We're also making existing software available. So Titley have a program called Insight. There's a number of software to do this. This is a frequency sonogram, again, of a bat call in the middle of the screen. And you can use this program to download metrics about the call, to write filters, to begin the process of allocating a call to a particular species. We've realized that the database is going to be so big that individuals won't be able to download it themselves without some effort. And so we're putting the software onto virtual desktops so people can analyze the data online. We're also developing a variety of analytical and statistical approaches. On the left here, we just have a simple discriminant functions where each color are the calls of a different species, and we can generate confidence regions where we are pretty confident that particular species fall, and then we can overlay calls of unknown species onto that and draw those dots out into a database to help us analyze those calls. There's also a whole lot of growth going on in image recognition techniques, and we're beginning to explore machine learning algorithms to make this process more useful as well. So I mentioned this builds on a recently established TERN program in Australia. On the left here, we just have to picture a map of Australia. The blue dots are existing TERN sites, and the colours, the reds, are areas of highest species bat diversity in Australia. And on the right, we have the same blue dots, TERN sites, but we've got just the distributions of some vulnerable species from Queensland. So by overlaying these maps, that helps us pick appropriate areas to get sort of the maximum return for our efforts to develop our core libraries. The last year has been very informative in Australia. We've collected over 23 million bat calls, representing 250 million individual call pulses. And through that process of looking at sounds, researchers have also detected a new species of bat just based on its call, and have gone out to those call locations and captured the bats to identify it. I just want to emphasize it's critically important that the call libraries have both identified and not identified calls. In Australia, and probably for some reason of Southeast Asia as well, we still don't know what all the calls are, so we're still at that basis. Data curation is key. It's 18 terabytes of data at the moment. We're planning to store this in Australian database at the moment, and Guru, who I noticed is one of your IT managers, he's in charge of that. So any questions for IT, we'll direct that straight to Guru. So look, I just wanted to finish there. A little bit fast, but I wanted to leave time in case there were any questions that people might have, which I'm more than happy to answer. And again, thank you for the support of ILTR. We're really looking forward to getting this up and running and potentially getting other countries involved as well.
So thank you very much, Simon. This was really quick and to the point, but it was still clear. That's not so easy. So thank you very much for this nice presentation. And this indeed leaves us with 4 minutes for questions. So if anyone in the audience would like to ask Simon a question, you're more than welcome to do so. I see a hand raised by Ellie.
Yes, I'm going to probably ask everyone the same question. Thank you very much. That was to the point and very clear. Even I understood it. What makes it an LTR project?
I think, well, I'm new to ILTR, so I hope I get this right. But from what I've learned, LTR is interested in recognizes the value of long-term monitoring and using that information for real-time information, among other things. And so it's relatively inexpensive to set up these. Detectors are less than $2,000. So it is relatively inexpensive to set up a device that will record every night, as long as the batteries are charged, and every couple of months you just go download it. Now, we all know that collecting data is the easy bit, but once we get this data, then that will improve our ability to be able to remotely survey species without ever having to capture the individual, which is pretty amazing capacity, I think.
Excellent, thank you. So yeah, so the long term is definitely the definition of LTR. That's on time. The space you showed already, the space is not required to make it an LTR. You can have a long-term monitoring in one site, but you showed that it can cover a large area. And is the scope of this research and monitoring that it will continue for many years and maybe even expand to other countries?
We would certainly hope so. I mean, the grant, they're small grants. We've managed to be able to purchase 6 detectors. So when we've chatted to a lot of people in Southeast Asia that would like to do it, but don't have access to detectors. So we'll set up 2 in each country, which again is, it's a fairly limited approach. But we already have support from Titley. My goal is that this would be a very nice seed project at the end of the year that's actually worked through the pros and cons, the difficulties of applying it in multiple countries. It would allow us to put up a very accurate and very reasonable budget that you could expand to however many countries that you wanted to, and obviously you need to justify that. But I think what we'll have at the end of the year is a lot more information about the bats and a very good knowledge of exactly what it would cost and what is required to expand it further, and hopefully a lot of interest and people willing to do that. I mean, the data is being made available to whoever wants it, and I think some of the people involved are students, and they may well be using it in their own studies, which is great.
Thank you.
You're welcome.
We should also add that this project has established clear contacts with a range of alternets in the region. So that's also very important, that it really connects across networks. Which was also one of the reasons why we selected this one. So is there one more question? And I should have someone coming in, should have told you that if you want to ask a question, the best thing is to use the reactions and raise hand, because we are so many, otherwise it will become difficult. Good, so then we thank you, Rob. We thank you, Simon, and for this talk. We thank you, Ellie, for the question, and then we go to the next one. The next speaker is Alex Siggers from Colorado State University in the USA, and talking about plants to soil, legacy effects of global change drivers. Alex, are you here? I think Alex has not joined, right? No, he has not. Okay, then we hope that he may join us later, and then we go to the next one. The next one would be Simone Moretti. And I see that Simone is here, and I kindly ask you to start with your presentation. It's about monitoring changes in Antarctic biogeochemistry of nitrogen. And we see again the presenter, yeah, so that you could go to the audience mode, please.
So hello, everybody. Just gonna try to share again on the other screen. Hopefully that will work better. Can you see right now?
Yeah.
Okay. So thank you all for this opportunity to present this project. So now we jumped a bit toward higher latitudes. And this project is about monitoring changes in Antarctic biogeochemistry of nitrogen. So this presentation will briefly introduce the Southern Ocean and the Circumantarctic region, and how can we use nitrogen isotope to to track Antarctic biogeochemistry. And then we will introduce sediment traps as long-term archives of biogeochemical changes, which will really be the core of this proposed research. And I will also briefly show some zooplankton delta 15N, so nitrogen isotopic analysis from some sample from these sediment traps as a preliminary study to show you what we can do and we can achieve, and finally show you a timeline of this project. So why studying the Southern Ocean? Well, the Southern Ocean is kind of unique in terms of the interplay between ocean circulation and biology. So deep water from the ocean interiors up well in these regions bring up a lot of nutrients and also a lot of carbon that eventually gets released into the atmosphere. And they do regulate the climate of the Earth in this process, as a lot of the CO2 eventually affect the radiative balance of the atmosphere. But also the Southern Ocean is a huge carbon sink for the anthropogenic carbon that we emit every day. So 50% of all the carbon that goes into the ocean is uptaken by the Southern Ocean. So this part of the ocean is really critical for global climate. But we also know that it's changing very fast. It's warming faster than the global average. We know that westerly winds, which are responsible for this upwelling, are intensifying, and we have coastal freshening that kind of affects the overturning rates of this deep water in the ocean interior. What we also don't know is that how will the biological pump respond to these global warming changes in the future. So the biological pump is that mechanism by which you take nutrients from the surface with primary production, and then you pump them into the ocean interior, whereas sequestering carbon that would otherwise end in the atmosphere. And we can use nutrients to track also the carbon cycle because the two are tightly linked. And as you can see in the next slide, you show the same section that you saw before, south to north section, but now depicts nitrate that is tightly mimicking what the CO2 is doing. And that is because the distribution of nitrate in the Southern Ocean is strongly affected by the degree of nutrient consumption by primary producers, so phytoplankton. So phytoplankton, when they assimilate the nitrate, they preferentially take nitrogen 14 in this nitrate because it's easier to assimilate. And therefore, they leave behind waters that are isotopically heavier, so to say. And this can be seen in the section below, which shows the distribution of the isotopic composition of nitrogen of this nitrate in the water. And as you move toward the north, toward area where waters are more fully depleted of nutrients by primary producer, the nitrate, delta 59, becomes heavier. And you can also see it in this plot here that shows that the more you consume nutrients, the more the residual nitrate it becomes heavy. But nitrogen isotopes are not just a tool for reconstructing the degree of nutrient consumption, they're also an established and well-known tool for the structure of the food web. So we know that the more you go up in the food web, the more enriched this time in 15N you are, because you tend to isotopically retain isotopically the heaviest isotope and excrete light isotope from your food source as part of your metabolic processes. So while we know that the nitrogen isotopes are a very established and powerful tool for reconstructing biogeochemical cycles, their applicability has been hindered because this organic matter doesn't preserve well back in time. So in fact, most of what is produced in biomass gets remineralized back to nitrate. And therefore, this information is lost. And if we want to really understand where we're going in the future, we need to also understand what changed in the past. And sediment traps provide us kind of a loophole to preserve some of this organic matter from the past so that we can study it today. So the sediment traps that have been deployed in the Southern Ocean as part of the long-term research, ecological research, have been going on since the early 1990s. And basically a sediment trap is nothing but a giant funnel that connects particulate organic matter falling from the surface of the ocean that eventually gets funneled into a carousel with many bottles that collects over the time of a year several particulates. And these bottles have spiked with like solutions such as formaldehyde that preserve organic matter from decay. And in this process, you can preserve organic matter that would have otherwise been lost. And this is just a picture I took from one of these samples from 1996. And you can see the incredible treasure of organism that you can still have very well preserved in one of these bottles. And the idea is that we study the isotopic composition of these organisms and diatoms, so primary producer and zooplankton, to reconstruct food web structure over time. So these are the sediment traps that we will propose to study, and basically is mooring B and A from the Ross Sea that is currently maintained by Italian Antarctic expedition that go there every year. And we have the PalmRes Long-Term Ecological Research Station, which was an American program. And the years that you see outlined here are the years for which we have obtained samples already. And therefore the years that we proposed to the ILTER Research Initiative. We are also working to get more samples from the PalmRes because this time series actually go back to 1992 and continues, I believe, to 2022. In addition, for the Ross Sea, we also have planned seawater sampling for these years that is taken in the same location as the mooring. So just to give you an idea of what is in this sediment trap, you see there is really a treasure of organic matter. For every year, you have these many bottles where you can see the beautiful progression of primary productivity in the austral summer and then the transition into the austral winter. So imagine that we have 3 decades overall that covers this high latitude region for which we have very little instrumental record with this resolution. And the beauty that we can extract from this organic matter phytoplankton and zooplankton. We can sieve this organic matter, we can use separation method, using density separation method or manual picking under a stereo microscope to separate individual zooplankton species and phytoplankton and perform chemical analysis of nitrogen in these. This is, for example, a test that we've been doing. We have been selecting one species of omnivore zooplankton, Metridia gerlachii. It's the most abundant copepod in the Antarctic coastline. And this has been separated by a collaborator in this project, Marco Grillo, which is a zooplankton expert in Italy. And we've been, each dot that you see here is an analysis on one individual that we've been performing at the Max Planck Institute. And we've been testing different preservation methods and how they affect the nitrogen isotope, because preservation method is a big discussion in the community or whether it affects your chemical analysis. And here we can see that ethanol or formaldehyde don't really have an impact. And in the sense that the analysis that we get from individual specimen, we see a variability that is much bigger than what you can explain with preservation method. And more importantly, the isotopic composition of this initial sample show that the signal is preserved because they are enriched with respect to the baseline of the food web, which is nitrate, delta 59. And this zooplankton is consistently heavier, suggesting that they are really recording a primary signal, not just an issue of preservation back in time. This is just a cartoon to outline to you what can be done at the end of this project if we put together.
Simone, sorry for interrupting, because 10 minutes are up, so we should relatively soon finish.
Finishing up, thank you. So basically this time series just show that we can monitor the structure of the food web with time and by measuring phytoplankton and zooplankton. And this is the potential of this project. And the timeline is basically stage 1, pre-funding work that has been done. We cataloged and collected all the samples. And now we are in stage 2 where we're analyzing different pool of organic matter and seawater nitrate. And then over the next year, we will analyze more diatom, hydrogen isotopic composition, and additional zooplankton species. And with this, I want to thank you for your attention and also thank you to ILTER for this opportunity. And I'm happy to take questions even though I'm a bit over time. Thank you.
Yeah, thank you very much. We will actually not take questions now because we really need to keep the time, and I don't want to go over because if Alex still appears, which may happen, then we would also give him a slot. Therefore, thank you very much. And we will move on to the next one. We will talk about TOPOGRASS, Microtopography Effect on Grassland Response to Extreme Drought. And it will be presented by Aniko Cseczerits of the Institute of Ecology and Botany in Hungary.
Maybe now, do you hear me?
Yeah.
Oh, thanks. And do you see my slides?
Yes, but not yet in presentation mode.
Okay. Okay. I try to sorry.
You see this little on the
but there is also a black black slide. Sorry.
In the red bar on the bottom, there's a little screen. If you click that.
Yeah, but it is
hidden. A bit further to the left.
Yeah, but it is hidden with the zoom. Zoom. Sorry, maybe this way. No, it is okay?
Nope.
Oh, not yet. Because I changed in the slide. Unfortunately, there is the zoom slide, this black one, and I cannot hidden this one. And this is
You can also use one of the controls in the top. You see the big screens there. I cannot really understand the Hungarian, but there will be something in a moment. Yes,
this is what I tried. Now you see the slides? Is it okay?
No, it's not.
Oh, sorry.
But if it doesn't work, you present like this, because we don't have time.
Really sorry. Then I start. I don't know why it is. Okay. So thank you very much for the possibilities. And yeah, I would like to present our works. As everywhere, also in our places, the climate change is a big problem, and it can realize in two types. There is a continuously changing in the means of the climate, so the average temperature change continuously, and there are more frequent extreme events like droughts or droughts. The study of these long-term changes and the effect of the extreme events needs really a long-term monitoring, and there is a ILTER site in Hungary, which is also a good object for these studies, as every ILTER site. This ILTER site is dominated by grasslands, which we think it's a little bit easier object as another more complex habitat, because it answers more quickly to this climate changing. And our ILTER site, the Kiskun ILTER site, is dominated by this grassland, so there are an ongoing monitoring research. There is a permanent meteorological field station, and these are the data of this field station. You can see the increasing mean annual temperature, and also you can see the changing annual precipitation. In the last year, in 2022, there was a dry year, and there, there was a extremely dry year. You can see here on the below figure the driest 2 months of the summer, and it shows that in the last year there was a really extremely dry in this area. As a result of this drought, there was an extreme dieback of the dominant grasses. There are two dominant grasses, Vesticovaginata and Stipa borystanica, and they died out in a large.
Sorry, Aniko.
Sorry, yes.
We only see the first slide. If you want us to see more slides, you have to scroll down.
Okay, I scrolled up already a money. I go back.
Yes,
okay.
Now you see the third one.
Okay.
The fifth one.
Okay, because it changed. You see now the second one?
Yeah.
And the third one?
Yep.
I thought about this third one. Maybe should I repeat it, or I can skip it?
No, because there's no time for that.
This,
okay.
As I just repeated, this is the meteorological station, data of the meteorological station of this ILTER site. And this is some pictures about the dieback of the dominant grasses. You see it? You see the fifth slide, yes?
Yes.
No.
Okay, thanks a lot.
6, yeah.
Yeah, and the aim of this project was the resampling of this monitoring area in order to see how the dieback occur exactly, and create a fine resolution microtopographic map of the study area, and analyzing the vegetation changes, both in long term and both in response to the extreme drought, and accessing especially that how the microtopography modify these changes. Here is some pictures where this site exists and can be found. This is in the middle of Central Europe in Hungary, and there is an open sandy grassland habitat. There is no management in this particular area since the 80s, and it was established 200 random plots in 2000, which were 4 by 4 meter large, and there was several resamplings. And yes, the last one we succeed to make in this year. Yes, here is the field group of the resamplings. There participated both senior and young researcher, so the young researcher can learn the detailed methodology of this resampling. And after the resampling, we put some plastic plates on every plot, So, and after, so the drones can recognize the exact places of these plots. And there was made a new aerial photos, a very detailed orthophotos. And not only orthophotos, but also a digital elevation map was made by this survey. Yes, and we looked for the pressure effect of the climate change. So we looked at the changes in time, and we looked also at the pulses effect of the climate change, actually on the effect of this extreme drought in 2022. And in this case, we can use the result of the drone survey. We can estimate very punctually the exact aspect, slope, and height of the plots. And also we can survey the amount of woody species in 10-meter radius from this digitation elevation, from this area photos. Some early results I would like to show you. For example, the vascular cover of the vascular plant species hardly decreased in this year. And not only the all cover decreased, but also the cover of the two dominant grass species decreased because of this drought. You can see here how does the life form changed in time. There was a decrease in the cover of the perennial species, but there was an increase in the cover of the annual species, which pattern occurs also earlier in the year 2004. You have to know that there was also a small drought event, and the pattern repeated itself. So it's interesting to see how the pattern repeats itself. And after this drought event, there was a regeneration process. We are waiting if there is also a new regeneration process or not. Here you can see the produced digitation elevation model by this new survey, which is a very detailed digital elevation model of the area. And there should be made some correction also in order to cut out methodologically the trees and shrubs in order to get the real soil layer. These maps can be used for the modeling, and I would like to show you the really first results of this modeling, and we found a significant interaction between the slopes and the amount of the woodiness. Firstly, if there is more wood, there is less dead grassland, so the wood somehow protect the grasses, but also there is less dead grass in the cases of more steep slopes. So on these steep slopes, even with less woodiness, there is less dead grass. So there are different places for surviving this grass species. And on the last slide, I would say, and show you the present state and the work plans. So we finished the field survey. We have a new detailed digital elevation model for this area, which was not there before. And this make it possible to make a more precise statistical analysis about this area. And this will be a work of MSc student. And this make also possibilities to analyze the relation between the diet biomass and the topography and woodiness, which will be a part of the PhD work of one of our students. And we would like to predict the position, exact position, the dieback, and we would like to study if it is the same as it was before. And so we will be able to identify the refugee places of this area, and we can see how to help to mitigate this habitat to the climate change. So I would like to say thank for the ILTER seed grant and also for the Kiskunság National Park Directorate, and thank you very much for your attention. And really sorry for these technical problems.
And we thank you, Aniko. In spite of the technical problems, this was a wonderful presentation, so it was very nice. We thank you very much. We will directly go to the next one, considering time, but if Alex does not show up, we will have a short question session for all talks at the very end. That is, if you have a question to Aniko, keep it in mind, and you can ask it in 10 minutes, because we will now get to Pedro Paulo de Ferreira from the University of Rio de Janeiro. And the talk is going to be on palm species responses to climate change.
Oh, hello everyone. I'm going to share
Hello.
the slides now.
Yeah,
Sorry,
please do.
can you hear me?
Yes, we hear you well.
Okay. Now you can see
Yeah,
the
but we still
my
see the
slides.
previous stuff.
We still see Aniko slides. I don't know how this is set up. Usually your slide should,
Maybe I stopped.
Yes, perfect. Now we see it.
So I will present to you about our research initiative called PalmRes, which we intend to investigate the responses of palms to climate change using a gradient of environmental conditions along a mountain elevation in the Atlantic forest. My name is Pedro, and I am a postdoctoral researcher at the Federal University of Rio de Janeiro, and a Brazilian long-term ecological research site called Central Fluminense Mosaic of Parks and Reserves. And I am representing a team of collaborators that is composed by professors from the Ecology department at the Federal and State Universities of Rio, experts in the topics of population ecology, palm ecology, distribution modeling, and climate change. And we also have researchers and students at various levels of postgraduate education, and most of them are members of the Plant Ecology Lab, conducting research on the same topics that I mentioned before. So our project will be carried out within a long-term ecological research site, which in Brazil we call PELD program. And in our case, it's the Central Fluminense Mosaic of Parks and Reserves, the MCF. The MCF site is a prime area for the conservation of Atlantic forest remnants and a wide range of endemic species, having recently been designated as priority area for palm conservation. And the MCF is also particularly important for investigating the effects of climate change on biodiversity, because it encompasses a gradient of environmental conditions across an altitude range from sea level to 1,500 meters. And additionally, palms are an interesting group to perform ecological and evolutionary studies due to their non-geographic distribution and high variation in species diversity and life forms. So understanding the responses of palms to environmental variation can infer about their climate vulnerability under climate change, especially endemic and endangered species such as Euterpedulis, the Jussara palm. Thus, our main objective is to investigate the impacts that climate change has already had over the last 10 years and the potential impacts expected for the next 30 years on the MCF palm communities. Specifically, we intend to assess the relative importance of assembly processes structuring palm communities at different spatial scales along the altitudinal elevation. In addition to evaluating the taxonomic and functional composition of palm communities, we will describe and characterize the sampling sites according to microclimatic and topographical factors. And we will also investigate how populations of the most abundant species behave in terms of demography, determining parameters such as population growth rates, vital rates, and elasticity. We will seek to understand palm responses to environmental variation that may make them vulnerable to climate change, as well as the effectiveness of protected areas for palm conservation. So between 2013 and 2014, 160 plots of variable area distributed from sea level to 1,400 meters of altitude were established. We used a modified variable area method for palm sampling that has been empirically proven to be effective to sample species with aggregated distributions. And in these plots, 12 palm species were identified, and more than 400 adult individuals were tagged. We also collected some microclimatic and topographical variables from the plots. And now we will come back to resample the same plots 10 years later. This is the map of our study area, where you can see the limits of the MCF, which covers around 300,000 hectares. The MCF is composed by a mosaic of fully protected and sustainably managed protected areas under state, federal, or private administration. And the numbers represent the largest and most important protected areas in the region and in the state of Rio de Janeiro, which are our sampling sites for palm community assessments. These sites are positioned along the altitude on a mountain elevation and consequently subject to different environmental conditions. And to understand how this environmental heterogeneity experienced by populations affects resource use strategies along environmental gradient, this set of functional traits are being measured for all palm species that occur in these plots. We have morphological and physiological traits measured at the leaf or the whole plant level, some of them related to photosynthesis performance, some are related to reproduction. Most of them we have already measured, and there are a few others that we still need to collect. To map climate change in the protected areas covered by MCF, models will be generated with climate variables under current and future conditions under 2 climate change scenarios. Maps with exposure values and the degree of vulnerability to climate change of MCF protected areas will be produced. This initiative is developed in partnership with the management of protected areas and continuous ongoing projects by long-term ecological research sites and postgraduate students in training. With the data that we will collect, we hope to verify how palm populations respond morphophysio and phenologically to the variation in environmental conditions, and thus identify demographic and functional traits of palm species that may be more vulnerable to local extinction. And we also expect to generate current distribution maps and projections in climate change scenarios for species, with emphasis on threatened species associated with higher elevation range. In addition to scientific papers we intend to publish, we also intend to produce press releases for scientific divulgation, a palm species identification guide to general public. So thank you everyone for listening, and to all the supporters of this research initiative.
Well then, thank you very much, Pedro, and thanks for keeping the time. This is very nice. So we now have some minutes left. We want to finish in time, but we still have some minutes left. Alexigers has still not appeared, so we will take some questions, and you can actually ask if you wish to any of the presenters. And if you want to ask, please raise your hand. Well, this used not to be the case, but if you can dare to raise your hand if you want to ask anything. They are all friendly people and will happily answer whatever is on your mind. So if that's not the case, then we will close our webinar. And what's next? So what's next is that our research initiatives will continue the work. They're all at the beginning, very early stage. So that's also why Pedro's presentation ended with expected results. And we will report on these initiatives through the website so you can follow what's happening. And there will, of course, be more research initiatives next year. And all the presentations will be made available to the coordinating committee members. So I ask that all the presenters, unless you have already uploaded your presentation, either upload them or you can also send them to us via email, so we have them. All others who have joined the webinar and are not coordinating committee members, we will share the presentations upon request. You simply write to office@ilter.network unless one of the presenters disagrees. Okay, so if you don't disagree, we will make them available upon request and for the coordinating committee members on our regular cloud. And in that case, I thank you all for presenting. I thank you for the effort you did to submit proposals to us, for the interest in our program. And again, also many thanks for Deshane for setting up this webinar. This was really interesting and I think a good learning opportunity for all of us. And when we do it next time, we will hopefully have Alex also with us. Then we close the webinar. And for the coordinating committee members, we see you tomorrow